Hi there, my name is Allie Barber, and I am a graduate student here at CU Boulder in the biochemistry program. And today I'm going to be talking to you about the project I'm working on in my first lab rotation in the Dr. Goodrich and Dr. Kugel lab. So first I want to talk about a major concept in biology called the central dogma. Basically, the central dogma outlines that DNA is going to be converted into RNA via a process called transcription, and RNA is converted into proteins, which are essential for life and cellular function, via a process called translation. So this lab is interested in different mechanisms of how transcription is regulated in the cell. So that's that first step in the central dogma, DNA converting into RNA. The focus of my project is to look specifically at something called B2 RNA. This RNA is found naturally in mouse cells, and it's part of a family of non-coding RNAs. Non-coding RNAs means that the sequence does not encode for a protein. While B2 RNA does not synthesize proteins, it still holds a very important role in cellular function, and it's been shown that that major function is transcription repression. Now, if transcription converts DNA into RNA, it's important to understand why the cell might benefit from repressing that process. The ultimate effect of transcription repression is gene regulation. So that means increasing or decreasing the levels of gene product. By gene product, I mean either RNA as a gene product of DNA or protein as a gene product of RNA. So transcription repression allows the cell to adapt to changes in external stimuli, such as a stressful environment or changes in energy source. And this keeps the cell from producing protein when it is not necessary, which can be very energetically costly. So one finding is that the levels of B2 RNA in the cell are greatly increased when the cell is introduced to stress. So this could be heat shock, viral infection, or DNA damage. So this is an example of transcription being repressed by B2 RNA to adapt to an external stimuli, in this case, a stressful environment. So how is this transcription repression achieved? The mechanism involves an enzyme called polymerase II. Polymerase II's function is to bind to DNA and aids it in transcribing into RNA. Dr. Goodrich and the lab has found that B2 RNA binds to polymerase II after heat shock, an example of stress, and it forms complexes that disallow the polymerase from attaching to DNA. Therefore, the transcription of that DNA to RNA does not occur. So, if the cell has responded to a stressful environment and repressed transcription, but now needs to continue synthesizing protein, how does the cell get back to normal transcription? One possible answer to that is the fact that B2 RNA can be extended, and that would be by adding about 18 nucleotides to the sequence. A discovered effect of this extension is that the structure of B2 RNA is destabilized. So if we look at an outline of this process, stress induces the production of B2 RNA, which represses transcription. The extension of B2 RNA causes the RNA to destabilize, making it more likely that it will degrade. And if it does degrade, the repression of transcription is lifted. So while the function of extended B2 RNA is not confirmed, one strong possibility based on this evidence is that the extension of B2 RNA is part of a regulatory system to stop the transcription repression. And this is vitally important so that B2 RNA stops repression transcription when it's no longer necessary and proteins can continue to be synthesized. So now I'm going to talk a little bit about the rationale of this project. Why study B2 RNA? So there's a human analog, which means similar molecule to B2 RNA, called ALU RNA. It is also a non-coding RNA, and it has similar transcription repression as B2 RNA. Additionally, ALU can also be extended in human cells with possibly similar regulatory effects. So there's a lot of similarities between these two RNAs. If we can characterize B2 RNA and develop an effective experimental model, we can use that to study non-coding RNAs in humans as well. So the work currently being done by graduate student Tom Rivas in the lab is to characterize the B2 non-coding RNA to learn a little bit more about its mechanisms for transcription repression. So we're looking at two major questions here. One is, where is B2 RNA actually localized in the cell? The answer to this question could provide insight on the binding mechanisms of B2 RNA with polymerase II, which remember is part of the transcription repression mechanism, but it can also show us any other binding partners that B2 RNA might have. Currently, I'm working on experiments in the lab to determine the rates of B2 degradation. So rapidly degrading RNAs typically signify that they have some kind of regulatory role. So studying the rates of B2 degradation could provide more information about its function in the mouse cell. So in review, we have B2 RNA, which is a non-coding type of RNA that likely plays a role in repressing transcription, which is the process that converts DNA to RNA. In optimizing experiments that characterize B2 RNA, we are developing a set of parameters in which we can study similar types of RNA in human cells. This will allow us to gain more insight into the regulation of transcription in human cells, which is a vital process for our cells to properly function. So thank you for your time, and I hope you learned something interesting.